So yeah, the COVID-19 pandemic. So these are data from the first month of the pandemic, and there are two types of data we can get from the epidemic data we can collect. One is about the change in the numbers of different infected hosts in the population. So here in different regions, but the total number of infected individuals at a given time is shown to fluctuate over time. We have some waves. We're all familiar with these waves, but there's a steady increase and some waves in fluctuations in the number of infected hosts. And the second kind of data we can have is evolutionary data, which is more about how the different genotypes or variants of the virus are changing over time. And here we have data from the United Kingdom and from India showing that in these different countries, you have different, at a given time, you have different genetic diversity of the viral population. So not when alpha strains was dominant in the UK, for instance, it was not as dominant in India. So, and what we see is that we have a succession of genotypes that reflect the change, the evolution change in the viral population over time. These are two different types of data. One is really ecological data, so you just go out there and you count number of plants, species, animals, viruses. It's really what ecologists do. And the other is more about kind of population genetics, look at what's happening in the viral population. And the goal is to try to couple the two, because during the COVID-19 pandemic, it was very clear that you couldn't ignore evolution while the epidemic was going on, because there were all these variants with different transmissibility of virulence or resistance to the vaccine that were emerging all the time during the epidemic of COVID. So these two outlooks, like the epidemiological outlook and the evolutionary outlook, separately they're well understood. We have a good mathematical theory to do epidemiology and to do evolution, pathogen evolution in particular, and so what we are going to do in the following is to try to couple the dynamics of these densities I and the frequencies of the different strain Fi, and see how we can get some understanding of how the ecology and evolution of pathogens into play. So I'm just showing you two books that reflect, for instance, that we have enough knowledge separately on epidemiology and evolution of the infectious disease. So I chose these two books because they are Dutch authors, of course, so it was a normal thing to do, but we have a very good understanding of mathematical epidemiology. This is really a huge field. And we have also a good understanding of how to model long-term virulence evolution. And that was all of this book that was published in the early 2000s, trying to model how infectious disease evolve on the long term. But for COVID, we need to look at things on the short term too, because everything happened very quickly during the COVID-19 pandemic. So epidemiology, the aim is to understand the interplay between the two processes, especially when the timescales of the two processes overlap. And in order to describe these joint dynamics, we have a lot of models that we could borrow from in evolutionary ecology. These are population genetics model, adaptive dynamics model, and quantitative genetics model. So the goal of this talk is to start with a brief overview of these methods, because maybe not everyone is familiar with them, and present them in an epidemiological context. And then I'm going to do two things. In the first part, I'm going to describe a methodological framework that attempts to bridge the gap between these approaches, and show it can help us shed light on the interplay between evolution and ecology on a short-term and long-term timescales. And then I'm going to show how population structure can affect the dynamics of host-pathogen interactions. And I'm going to use COVID-19 vaccination as a motivating example. So, as an introduction, an overview. So imagine we have a host-pathogen interaction. And so there's one host, like the human population, and there's a pathogen, which may be the virus. And going to have the host population going to be infected by several strains of the virus. Okay, and a simple multi-strain SIR model in that case is as follows: you just have a dynamical equation for the dynamics of the susceptible host and for the dynamics of the infected host by strain I. And what is going to be important is, if you look at this dynamic, is that I'm going to introduce a function R, which I'm going to call the fitness function. And this function depends on the traits of interest, so maybe virulence, pathogen virulence, if we are interested in pathogen virulence. It can be any trait. And on the environment experienced by the pathogen, and here it's going to be simply the density of susceptible hosts. And what this fitness function tells us is that the density of strain I is going to increase in the population if, due to the process of transmission, which occurs if there's enough susceptible hosts and the rate of transmission is beta. And there's also a mortality process, which is due to virulence and which is going to decrease the density of host populations. Density of hosts infected by the strain I. So this is a very standard SIR model, but just we have a strain-specific trait that describes the pathogen viral traits. So this is the starting point. And what we do in epidemiology, and we typically we're going to average over all strains and ignore the genetic variation. We can sum all the infected case, and we get an equation for the dynamics of the total density of infected hosts. And we can express this simply in terms of a mean growth rate R bar, which is that's the average of the growth rate of the different strain in the population. So what ecologists are going to measure, if they measure the growth rate of the infected host population, is this average growth rate, and they don't necessarily have access to the genetic data underneath. Okay. To calculate this average, we need the frequency of type I, Fi, which is simply the density of infected hosts by strain I divided by the total density of infected hosts. And so you see here that we have these two variables I was talking about, the density of infected hosts and the frequency of strain I. Okay, and the goal is to try to see how these two interact. What's important is that they are necessarily coupled because the total growth of the population depends, of course, on the fitness of each pathogen strain, but also the discrete trait distributions of the FIs in the viral population. So the goal is to couple this ecological equation with an equation describing the changing frequencies. And one way to do that is just to calculate the dynamics of the change of frequency. And that's what population genetics does. Okay, population genetics assume that the trait is fixed for each strain, and then just looked at the change in frequency. And often we get an equation of this type, which is that the change in frequency of strain I will increase if the fitness of the strain I is greater than the mean growth rate in the population. And this is the replicator equation that maybe you're familiar with. But it's the standard equation in population genetics. The difference here is that in contrast to many population genetics models, now the fitness is going to depend on a dynamic variable, which is the density of susceptible hosts. So it's not a constant parameter as in population genetics. And with two pathogen types, we have this equation, which is simply that the change of the mutant, M, the frequency of the mutant, is going to be given by a genetic variance term times a selection coefficient, which is just the difference between the growth rate of the mutant and the growth rate of the wild type. So this is all very standard in population genetics. It can be applied to this model. And in fact, I'm going to show you in part 2 something which is very close to this framework. But there are other ways to model evolution. And one of these is adaptive dynamics, which has a long history with the Netherlands. The idea here is to look at long-term evolution. So in population genetics, you look at short-term evolution, the change in frequency. And now in epidemics, you are interested in how the mean trait change over a long time scale and whether you're going to get to a kind of equilibrium stable strategy, okay, the ESS of evolutionary game theory. And to do that, you assume something which is that the mutants are rare, so they arise at low frequency in a resident population that has already reached its ecological structure. And then you compute mutant success using this fitness function that you evaluate it for the mutant trait in the resident population at equilibrium. So the S with a hat is the density of susceptible hosts on the equilibrium, for instance, if that's an equilibrium, or maybe you compute it on an ecological structure, which is a periodic or limit cycle, if you have periodic dynamics. The important thing that you have this kind of separation of time scales between ecology and evolution. And then we have a whole toolbox, which I'm going to review because it's going to be important later. You have the fact that the direction of selections is given by the selection gradient, which is the slope of the fitness function when the mutant is close to the resident. And that has this form, so it's given by the slope of the transmission curve times the density of equilibrium of susceptible host minus the slope of the mortality function. So it is what we call the local slope of the fitness landscape. And then you can identify the potential evershed endpoints by looking at when this selection gradient vanish, okay? And then you can investigate the evershed stability of these evershed singularities. And for this, you need to look at the curvature of the fitness landscape. That's all standard theory for the inactive dynamics. I'm not going to go more into detail, but there's another branch of another theoretical framework in evershed ecology, which is quantitative genetics, and does something a bit different. Because it's supposed now that we have a trait Z in the population, which has a continuous distribution. And the goal is to know how this trait distribution changes over time. And because it's a difficult problem in general, the idea is to look at the change in the mean traits, so like the dynamics of moments, like the mean trait or the variance. And in that case, the dynamics of the mean trait are given by another equation, which is not the price equation in evershed ecology, and says that the change in mean trait is given by the covariance between the trait and the fitness of the associated with each strain. So we have a dynamic equation for the mean trait, and it has to be coupled with a dynamical equation for the densities of susceptible and infected hosts. So that's fine, but it's often not easy to handle. And so often we make another approximation, and we assume that the distribution is actually clustered around the mean. And so we can do a Taylor approximation, and we get something that says that the mean trait change. And again, we have a product of a variance and a selection gradient term. So we need variance for selection to act, and then the direction of selection is given by your selection gradient, which has the same form as before, as in the adaptive dynamics model, except that now we look at the instantaneous density of susceptible hosts and not the equilibrium density. Yep.
How in general you can like compute the evolution of this phi, so the distribution of the check, if satisfy some Fokker-Planck equation or what in general for the phi?
For the phi, so in quantitative genetics, there are many derivations where you assume that you have a Gaussian distribution, and so you got
to how that distribution evolve over time, because it depends also on t. Even if Gaussian,
it depends also on
t.
On t.
You have two
yes.
variables, z and t. So it
yes,
over time.
but that's why we look at the moments, and then we have a hierarchy of moments, and then we close the system usually by assuming we just track the mean and variance. That's what.
You don't need to solve explicitly an equation for time.
You don't need to solve it explicitly, but you can do it sometimes. But often you have some simplifying assumptions that allow you to close the kind of higher dimensional problem. So I'm going to explain this a bit in the other framework later, so maybe it will make more sense. This is more like an overview of where we stand with. Okay, so the idea is that I'm going to try to make a connection between these different models and see how we can shed light on a particular problem, which is what happens in an epidemic when you have evolution and epidemiology going on at the same time. And so for this, I'm I'm going to highlight the strengths and limitations of different frameworks. So the population genetics models, they can be used to explore this kind of rapid evolution dynamics that I'm interested in to explore COVID-19 dynamics, for instance. But they are less suited to study long-term dynamics because there's no standing variation and no recurrent mutation that fuel the genetic variance in the population. So what we get often is that we have a mutant and resident, and one of them is going to go to extinction, and the other is going to go to fixation, and then we have a monomorphic population, and no evolution can happen because we have no variation. So we have to add other processes, and it's not always easy to look at the long-term evolution in that case. Adaptive dynamics is better suited to look at long-term evolution and take account the environmental feedback, so the fact that the density of susceptible hosts is going to change during the epidemic, but it assumes that evolution is mutation limited, so that you assume some very specific assumptions on mutation, which is that they are rare, and often you assume that they have small phenotypic effects. So if you look at COVID-19, it doesn't match the assumptions we have where we had a lot of mutations coming in at a given time and a lot of variation. And quantitative genetics is better to look at rapid evolution dynamics, but you have these assumptions that typically in most models, you assume that the trait distribution is unimodal, narrow, and that there's a constant variance in the population, which are strong assumptions that do not match often what we have, where we could have multimodal distributions, or we could have populations with a large distributions with a large variance or variance that change over time. And so there's an approach that was first suggested by Sasaki and Dickman like 15 years ago, and that can be used to bridge the gaps between these different frameworks. And the idea is to start from the trait distribution as in quantitative genetics, but then to decompose it into a mixture of morph distributions. So, for instance, you assume that you have this distribution is the sum of two morph distributions, each with a specific mean. So this is the mean of morph 1, this is the mean of morph 2. Then we have a standard variation for each of these morphs, and the heights of the peaks basically are the frequency of each morph in the population. So basically, it allows you to handle multimodal distributions, but use some tools from quantitative genetics to describe what's happening for each morph. And when you do that, you can characterize each morph by its position, which is given by the morph means, its relative abundance, which is given by the morph frequency Fi, and the width of the morph, which is given by the morph standard deviation. And what we're going to do is to look at how these different characteristics change over time. Okay. And what we do for that is we assume that within each morph, we assume that the variance is narrow. Okay, so basically the multimodal distribution will look like maybe a sum of peaks, not necessarily direct masses, but we are going to assume that the morph are well characterized, and so that in a given morph, the trait is always close to the morph mean Zi. Okay, and so that means that the morph variants are small, and then can do Taylor approximations of the fitness function around the mean of each morph and derive some equations. The first equation we can derive is the dynamics of the ecological densities. Okay, and what we obtain if we do that is that we have two terms. First term corresponds exactly the expression of mean growth rate that I showed before. Okay, so it corresponds to the average of the fitness functions, but the fitness functions now are evaluated at the morph mean only. So that's an approximation of the fitness function, the first zero order approximation of the fitness function. Yeah, just an aside, I'm dropping the dependency on S for the moment, but it's still there. It's just to simplify the notation. Okay. And then we have a second term, and this second term it depends on the curvature of the fitness function near the morph mean. on the variance of the morph, on the frequency on the morph, and then you average all that on all strains. And basically, it captures the fact that because there are deviations from the mean trait, the mean growth rate is going to decrease. This is known as the genetic load in quantitative genetics, and this is captured by this equation. Okay, so this gives us an equation for the density of the total density of infected host, and to leading order, this is everything happens as if we approximate the fitness by what's happening at a morph mean. Okay, but it also tells us how genetic variation affects the mean population growth rate, which is some new information compared to what we had before. Then we can write the dynamics of morph frequencies, and again, we will cover the replicator equation. So, but we, with the fitness function, they've rated the morph means as an approximation. Okay, the difference now is that the morph means are not fixed. Okay, these are variables, and they are going to change according to another equation. Okay, and this equation they tell us how the heights of the peaks of the distribution change over time. Okay, so we have our multimodal distribution. Maybe one peak is going to 0. That means that the frequency of the morph is going to 0. Maybe the two peaks are going to go to 1.5 or something, and then we'll have a bimodal distribution. We can track this dynamics. So now for the dynamics of the morph means, we need to calculate approximate the dynamics of the morph mean, and we recover an equation that is just a morph-specific version of the quantitative genetics equation. That is, the morph mean change depends is scaled by the variance of the morph and by the slope of the fitness function near the morph mean. Okay, so for each morph, we have this equation. That means that for each morph, we know how the mean trait is going to move, and so the equation tells us in which direction the modes of the trait distribution move. And then we have the dynamics of morph variances, which I've not shown before, because most quantitative genetics models assume that the variance is constant, and so they discard the dynamic. But here we can explicitly track the change in variances, and we can show that the dynamics of variance is given by different things. It depends on the amount of genetic variation in the population, BI. It depends on the degree of presence of outliers, which is measured by the kurtosis of the morph distributions, chi, and it depends on the curvature of the fitness function at the morph mean. And so we can use this equation to track how the variance of a given morph changes. And so what this equation will tell us if, depending on whether the variance shrinks or increases, is going to tell us if we have stabilizing or disruptive selection around each peak of the distribution. So if you imagine that you have one peak in your distribution, if the variance shrinks, it means that selection is stabilizing, so it's going to keep the distribution around that the mean of the distribution. But if its variance is increasing, the distribution is going to spread, and potentially this is a signature of disruptive selection.
And the second derivative there, that's negative?
So it depends on your model. In the ISRR model that I show, yeah, it's negative because you often assume that the transmission is a concave function of the trait.
Because in one of the earlier slides you said that actually a term involving that second derivative decreases the mean growth rate. So I inferred from there that it actually was a negative.
Yeah, okay. So it can be positive, but in many models of quantitative genetics, they assume that it's you only have a Gaussian fitness function, and so when you move away from the optimum, it's going to decrease your growth rate, and so the effect will be negative. Okay. But you could imagine in a more different model that could be a positive effect too. Yeah. I mean, often the link with quantitative genetics tells you that it should be negative, and often in many models where you have an ESS, that's what what's going to happen, because you when you go away from the to decrease your growth rate. But so yeah, in the model I'm going to study is going to be selection will be stabilizing on each morph. It's not a generality, but.
And the kurtosis, how do you get to the kurtosis?
How do you?
So
the one assumption there is underlying assumption that I have not explicitly that the distribution is symmetric around each morph to simplify. So the third moments are 0, but then there's still in the derivation you still get the fourth moment coming in, and what's going to happen is that often if you use a Gaussian distribution, this can be approximated in function of the variance, and so the fourth moment disappears. But in general, it's going to depend on the fourth moment, and the fourth moment dynamics is going to depend on all the other moments, and you need to make some closure assumptions to close the model.
So, but from my understanding, I mean, you have talked about quantitative genetics, which also gives rise to a set of equations for various models that you can also close off actually at the variance, right? You can include the variance and then close off at the kurtosis.
Yeah.
This is basically adding systems of equations for different morphs to capture different.
Exactly. Yeah. So if you have only one morph, you recover quantitative genetics. But at least you recover like Barton and Turelli, this kind of work where they were interested in moments. But then people lost interest in that, and they say, well, variance is just going to be constant. And maybe for many models of sexual reproduction, that's okay, because of sexual reproduction maintaining variance and so on. But so most of the applications I know of for quantitative genetics, they assume that variance is constant, and you just look at the direction of the mean.
Yeah.
So, but if we are interested in branching or things like that, you can't just ignore the variance, of course. So you need to do something else. Okay, yeah. So that's what we were discussing. The system is unclosed, of course, and so often what we can do is do a Gaussian approximation to close the systems. In which case, we have a very simple equation for the variance, which depends just on the curvature of the fitness function and the square of the variance. But you have other moment closure approximations that can be used. In fact, for the beta distribution, for instance, there has been some applications in ecology. Okay, so that's the setup. You may be wondering why it's relevant for COVID. So if I write all this in a kind of simple as susceptible infected host pathogen model, I have this system of equation. So there's an equation for the total density of infected hosts. It's going to depend on the frequency and the morph of means. So sorry, the frequency and the means of each morph. And then you have an equation for the frequency of the different morph. Again, it's going to depend on the frequencies and on the means. And then you have an equation for the morph means. Depends on the variance and the morph means. An equation for the variance of each morph, which depends on the variance and the morph means, and together with an equation for the density of susceptibles. So it may look like a complicated set of equations, but the idea that you can use that to get some insights on various phenomena at different timescales. And one of the things that is interesting also is that this integrates a bit the different framework we have in evolutionary ecology, and depending, we can make different assumptions to recover limiting cases. For instance, if we have a unimodal distribution, we recover quantitative genetics, as we just discussed. If we assume that the traits of the species or the morph are fixed, we recover traditional ecology or population genetics, depending if we interpret the equations as equations describing species or morph. And if you assume that you have vanishingly small more variance and rare mutations, you get back to adaptive dynamics. And the interesting thing also that you can integrate fast evolution processes and slow evolution processes in the same framework. What I mean by that adaptive dynamics is often assuming slow evolution. So you look at the long-term evolution using mutation-limited evolution. And population genetics and quantitative genetics, they look at the short-term dynamics, okay? Oligomorphic dynamics allow you to move in between these two scales, and that's what I'm going to show in this application. And so the idea is to use this to, and first thing we need to be aware of is that this naturally gives rise to a separation of time scale, okay? Because our epsilon parameter is small and the variance are scaled by epsilon. So we have some fast variables and some slow variables in this system. The fast variables are the total density, okay, of infected hosts and the frequency, so the peaks of the distribution. And the slow frequencies are the variance and means, so the width and positions of each morph. And we can use this separation of time scale to make some analytical progress. But we can also do something else, which is looking at the population level mean traits. So in the distribution, we have different morphs with means Zi and frequencies Fi. And of course, if we calculate the average trait in the population is going to sit, maybe if you have diagonal distribution is going to sit in between. And this is, we can calculate this mean trait like that, okay? And if we do that, of course, the change in mean trait is given by two components, one giving the changes in frequencies, so the changes in peak heights, and one giving the changes in peak positions, so the change in the morph means. And these represent different kind of processes at different time scale. And what is interesting is that if we look at the two morph models, so we have a wild type and a mutant, we can write the change in the mean trait as follows. So there's two components, a blue component and a green component. The blue components comes from the change in the frequencies, so the change in the height of the peaks. And the green component comes from the change in the mean traits, okay? And what is interesting there is that the blue component will dominate if the two morphs are very different. So that means the selection is strong, okay? The trait of the mutant is very different from the trait of the wild-type morph. And in that case, we have fast evolution, and the equation looks like population genetics, okay? We have a variance term, and then we have a term delta R, which is simply the difference in the growth rate of the mutant and the growth rate of the wild type, approximated by assuming that all the mutant and wild type are very clustered around their means, okay? Lead you in the direction of increasing your growth rate, basically. If the two morphs are similar, then this is the second line is the one that's going to probably dominate. And in that case, we have a slower evolution, like in adaptive dynamics and also reminiscent of quantitative genetics, where evolution is scaled by the morph variance and also driven by the slope of the fitness function. So in one case, we have what matters is which strain has the higher R, and in the other case, what matters for evolution is the slope of the fitness function, which is not the same. And now, my example would be to look at the evolution of pathogen virulence in this simple SI model, where the fitness function is just given by beta alpha times the density of susceptible hosts minus a mortality term plus a virulence term. And we're going to look at the evolution of this alpha variable, which is a virulence. We assume that there's a trade-off between transmission virulence. So basically, if you increase alpha, you're also going to increase beta, but there will be a saturating, the curve is going to saturate at some point, so you won't be able to reach as many benefits from increasing virulence after some time. We're going to start with a high density of susceptible hosts, like at the start of the COVID pandemic. We're going to start with the low density of infected hosts. Again, it's really like in the COVID-19 pandemic. And we are going to assume different distribution of virulence. So we're going to assume that we have two morphs, and they will have the multimodal distribution of these two morphs will be different in different simulation. And first, I want to start with an experimental example, which were done by some colleagues in the bacteriophage interaction, where they show that on the top panel here, we have prevalence. So they start with a low density of infected hosts, and you have a rise to a high level of pathogen prevalence after a short while. So this is an epidemic, okay? And they show that there are two strains that they compete, a virulent and a non-virulent strain. And you show that during this epidemic, you have a transient increase in virulence, so the frequency of the virulent host increases, and then decreases once we reach the higher level of prevalence. And we're going to see that the model allows us to understand why this is happening. So I'll go back to my system.
So
Yeah.
Can you go back to that?
Yes.
So, what is, this is virulent over non-virulent ratio. B is the virulent over non-virulent ratio. What? I don't.
Yeah, it's the ratio of the frequencies of
Okay.
the virulent and non-virulent phage in different compartments, the provirus, when the phage is integrated, and the free viruses.
And what is the blue line?
The blue line is another experiment where they started with 100% prevalence, and
Okay.
where they show that the selection pressure is completely different.
Okay.
And that's just because in one case you have a lot of susceptible hosts,
Yeah.
so there are a lot of benefits to, and in the other case you have no susceptible hosts, so you have only the cost of virulence. So that's two predictions of the model. I was only focusing on the first one, but that's what the blue line is about. So if we look at the dynamics of mean virulence, okay, remember this is the same thing I showed before. This is the delta R I was talking about, and this is the gradient terms that I was showing before. So the blue, again, the blue is giving the change in the heights of the distribution. The green is giving the change in the mean traits. And what's interesting, they can look at two limit cases. One where the variances are small and negligible. In that case, selection is going to favor the morph with the largest growth rate. So that means that this is going to be positive. And everything happens as if evolution was maximizing the R function. Okay, so the strength, the largest growth rate is favored. And one way to have the higher growth rate is to increase virulence. And if the two morphs are very close, okay, and this line can be neglected, we're going to have a slow evolution of the mean trait in a direction that is given by this gradient. And in fact, if the two morphs are very close, you can this one is equal to this one, so you only have one gradient that is driving you towards an equilibrium. The ST is a function of time, but can be approximated as a quasi-equilibrium approximation like that. And this brings us to an evolutionary endpoint where people from theoretical ecology will recognize a non-general value theorem. And that just means that we're going to maximize the R naught, so the basic reproduction number of the pathogen. So in one case, we're going to select for a large R. In other case, we're going to select for a large R naught. And that's two different regimes, it's two different selection regimes. Some pictures going to illustrate what I have in mind a bit better. So here we start with two morphs, but you see that our distribution are overlapping, okay? And so in that case, we have, if we look at this graph, which is here we have the density of susceptible hosts and here the mean virulence. You have a very fast convergence to the quasi-equilibrium, and then a slow convergence to the ESH, which is given by the maximization of the R naught. Okay, so if you look at the slow time scale, that's what you have. You have a convergence to the steady state on the short time scale, so the quasi-equilibrium on the short time scale, and then a slow decline towards the ESS. But if you assume that the morphs are slightly apart, then you start to have a bit of wiggle around this trajectory. So, and you see that you start having a very transient selection for higher virulence at the start of the epidemic. Okay, but then on the long time scale, you just converge to the ESS that's the same qualitative behavior. But now, if you increase the phenotypic difference between the two variants, what you have is a completely different feedback between ecology and evolution, because what you get is this kind of weird dynamics in the phase plane. And basically what you have is that you have, while the dynamics, the epidemics is still ongoing, you have a transient selection for higher virulence, and then the virulent strain is completely counter-selected once you reach the more steady state, and basically you have we select for a mean virulence which is below the optimal virulence calculated by maximizing R naught. But then over a long time scale, you're going to steadily increase towards this ESS. Okay, so qualitatively, it's completely different, because from a virulence management perspective, it's not the same as saying that the virulence of the pathogen is going to decrease steadily, or to say we're going to have a massive outbreak with a virulent pathogen and then it's going to die out, and because then the cost in terms of like human life, for instance, is completely different in the two scenarios. So you need to be able to understand a bit more carefully what is the transient dynamics of these of the pathogens if you introduce some public health measures, for instance.
And,
Yeah.
In all simulations, were the frequencies of the two types the same at the start?
I think so. Yeah.
So it doesn't depend. It doesn't depend on the frequency of the two morphs initially, or is this?
I think probably it could. I don't remember the details, because it was a couple of years ago that I did these simulations. But I think this is an accurate description. It's not a toy representation. It's really what the distribution we're looking at. Just the scale is a bit. Yeah. Okay, so what's happening is basically we start with an initial distribution, which is, I think, frequent like this, with the mean right here. And we have first a fast evolution regime where basically the heights of the distribution are changing, and so the virulent strain is okay. It's not. It should be in the reverse, but the virulent strain is selected, and then once we have competitive exclusion of the low virulent strains, we have of the high virulent strain. Sorry, we have a slow evolution regime that brings you to an ESS. And so basically this is pretty much what we have observed in the experimental evolution. And this also explains why in a real epidemic we have some transient selection for more transmissible or more virulent pathogens, because if there are plenty of susceptible hosts, there's a strong selection for to increase transmission, either by increasing transmission or by increasing virulence, which in turn increase transmission. I'm going to skip that because I have a second part. I don't know how I'm doing with time. That's fine. The second part is shorter, but just I want to summarize the first part. So the first part was, my goal was to try to show a kind of framework that first, I think is important for a better dialogue between different theoretical schools, because the problem we have in ecology, I don't know if it's in the other field, that some people are doing quantitative genetics, some people are doing adaptive dynamics, some people are doing ecology, and often it's not always possible to discuss with using the same tool. So here we have a kind of tool that can be used to put the pieces together. And I think there's potential with this tool to be able to be a bit more precise about how eco-dynamics work across different timescales, and perhaps to provide testable predictions on timescales that are adequate for experimental empirical studies, which is not always possible with long-term evolutionary predictions. This is joint work with Akira Sasaki and Mike Booth. The second part, which is a bit shorter, is I'm going to go back to COVID-19 pandemic and to ask a question about vaccination. And the idea is to show that we need sometimes to take into account the structure of the host population, because host populations are structured, they have age structure, they have immune status that is different between individuals. The number of contacts between individuals may vary. And so this creates a whole complicated landscape for the pathogens to evolve in. And from the pathogens point of view, if you have different hosts, they correspond to different habitats with different qualities, okay? Good hosts and bad hosts. The good hosts are those that are completely vulnerable. The bad hosts are those that are vaccinated, for instance. And we can ask, how can this heterogeneity affect the spread of different pathogen variants? The example I'm going to use is example of vaccination in COVID-19. This is work we did in the early days of the pandemic, when it was clear that the disease parameters were different across hosts, because there was a clear age pattern on disease severity. Some hosts are more contacts or could transmit more than others. And there was also large variability in disease severity independently of age. And after vaccination rollout, we had also some people were vaccinated, some were not vaccinated. We had different vaccine types, one, 2, or 3 doses, if you remember, that were affecting the vaccination status of the individuals. And at the time we did this work, we were wondering whether the recommendation we had, which was we need to target all people because they are more vulnerable, was the best from an evolutionary point of view. The idea is that COVID-19 vaccines were imperfect, and they still are in the sense that vaccinated individuals, they still transmit. Okay, they transmit at a lower rate, but they still transmit. And previous theory tells us that this is not necessarily good from an evolutionary point of view, because it means that the vaccine is leaky, and then it will select for maybe higher virulence or vaccine escape and so on. So we wanted to know, when we look at these heterogeneous populations, the fact that hosts have different relative qualities of pathogens was going to affect the speed of adaptation. Okay, so we were looking for a deterministic description of the change in variant frequency in this heterogeneous population to have a measure of the speed of our adaptation to the vaccine. So if you remember our population genetics framework before, so if we have a mutant in a resident population, so a vaccine-adapted variant, the frequency of the variant will change as follows. So now I'm using PM is a FM, but that's the same idea. The frequency of the mutant change. There's a variance term, there's a selection gradient term. Okay, this is standard population genetics, standard differential theory, really. And the problem is to compute the selection coefficient. And to take into account the difference in host qualities. And to do that, we can use a weak selection approximation. So we consider mutation to have a small effect. Okay, so E is going to be the trait, the escape trait of the virus, and we have an escape trait for the mutant and for the resident. And epsilon, the difference between the two is assumed to be small. And then we can actually approximate the selection gradient at time t, like this, as a product of first there's a matrix which is the perturbation of the matrix that collects all the effects of the mutation on pathogen transmission and survival. Okay, so that kind of the transition matrix of for the mutant pathogen. Then we have a vector of class frequencies that tells us what fraction of the pathogen population is in a given class at time t. That's a measure of the quantity of the different host classes. And then we have the vector of repetitive values. It's going to tell us how much the population, how much a pathogen in a given class at time t contributes to the future of the pathogen population. That's a measure of the quality. So maybe I should remove that. Quality of the host class for the virus. So this is
what is your E?
The E is the escape trait. So that means that we're looking at a vaccine that can escape, a pathogen that can escape the vaccine.
Right, so it's kind of a probability that.
So I don't really how do model. I don't remember if it was a probability, but it's yeah, maybe it's scale between 0 and 1, and you can. But it's how much. so the vaccine is reducing, for instance, the susceptibility to the disease, and this trait is going to make this reduction less efficient.
Yes.
So there's kind of we have so the susceptibility is a function of that trait.
Yeah.
So when that trait increases, I think it means that we have higher susceptibility to the disease, so the vaccine is less efficient.
And your RM, is that?
So it means that if you look at the densities of the pathogen in the different classes, you can write this in matrix form, and the transition matrix is RM for this pathogen strain. So compared to classical theory on classical population, the difference here is that we make all this dependent on T because we want to follow the change over an epidemic. Okay, so that means that the concept of reproductive value here is a dynamic one, and I'm going to talk about it later. But first, just to set up the ideas, the application is the following. We assume that the host population can be partitioned into two classes: hosts that have a low number of contacts, but they are more vulnerable to the disease, and hosts that have a high number of contacts, but they are less vulnerable to the disease. And basically, it was kind of a rough approximation of dichotomy because young people were working and had a lot of contacts, but were less sensitive to the disease, and old people who were maybe more at home because of all the restrictions, but they are also more sensible, more vulnerable to the disease. So it was back in 21 when we did that. So since then, the situation has changed with regard to vaccination. But at the time, the question we were asking was really, was it better to target the host with a high number of contacts or the host with a low number of contacts? So in total, we have 4 different host classes. The hosts that have low contacts and are not vaccinated, the hosts who have low contacts but are vaccinated, and the same for the hosts with a lot of contacts.
So, sorry to ask this.
No, it's fine.
More vulnerable to the disease, they spread it with the same rate as the ones that are less vulnerable to the disease. Is the variation captured by the number of contacts? The variation in transmission.
So I'm going to show you, I think, I'm going to explain this when I show the life cycle to be clear. So the question is, again, which individual should we vaccinate? And the fact that most studies look at that from an epidemiological point of view, so they ask, who should we vaccinate to get the number of infected cases lower? And the answer is actually not obvious when you have only epidemiology. So there are studies saying you need to vaccinate the old people, some studies saying we need to vaccinate the young people, and depending on the assumptions, and depending also on which metrics you seek to minimize, the answer is not unique. So already the problem is complicated from an epidemiological point of view, but our aim was to also look at this problem from an evolutionary point of view. So if we have different vaccination strategies, are they going to speed up or slow down the spread of the vaccine-adapted variant? So the model we have was that for the model, the host, we have low contacts and high risk, which are with an L. L is low contacts. We have this SIR model, which is adapted for COVID. So the susceptible host can be infected, and then they leave the infected compartment, and they can either recover or die. Okay, but they all leave at the same rate the infected compartment. And then we have, when you're vaccinated, this is the force of infection, and it's reduced by the effect of the vaccine on the susceptibility of the disease. And this susceptibility is modulated by the escape strain, the escape trait of the pathogen. And this is the vaccination rate, okay, of the rate at which the host become vaccinated. And we have the same picture for the host with high contacts, but the difference now is that we have also a parameter M that measures inflates the number of the force of infection of this category, and that has kind of a rough description of having more contacts. Okay, so this is a parameter that is greater than 1, and that captures the fact that this is a class with higher transmission overall. Again, there's an effect on the susceptibility for the vaccine, but otherwise the dynamics are pretty similar, okay, between the two compartments. And so what we do then is make some assumption also on the mortality rates, okay? And so we assume that the hosts with high contacts are less vulnerable to the disease, so they die less. And if you're vaccinated, also you have a lower mortality in high host than in low contacts host.
The mortalities of the vaccinated versus the unvaccinated?
That's a good question. I was thinking, I haven't put that, we did that in the paper. I don't remember what we did, but I will check for you.
and also this factor n that scales the number of contacts. Does that also scale your force of infection, that you have there? Because
it's just in front, of the.
Yeah. So the more contacts that these hosts have, they have it among each other and not with low contact hosts. Because you could imagine that if mixing is more, if a group starts mixing more, they also mix with.
Yeah, but they do, because the force of infection is it's a function of all the when you have transmission, you have contacts with everybody in the population.
So I still think that somehow it should also increase the force of infection for hosts with low contact.
Yeah, I see what you mean. I think, it's true. We didn't model this like this, but it could be interesting to pick this up. Okay. So we have an effect of the vaccine trait, the pathogen trait on the susceptibility and the transmissibility. So the transmissibility effect is hidden in the force of infection, and basically we're going to sum up everything by a trait E that captures the total escape trait of the pathogen. And what we do is we can use a quasi-equilibrium approximation to calculate the replicative values, okay, of which captures the contribution to the future population of a pathogen that lands in class L at time t. And you have these expressions. I'm not going to go into the details, but they depend on the transmission rate, which depends on the form of control that you can impose to the population. That's a measure of lockdown policies, for instance. It depends on the total growth rate of the epidemic. So whether the epidemic is increasing or decreasing, the result will be different. And it depends on the effective density of susceptible hosts. That means that it depends on how much susceptible the pathogen can effectively infect, depending on the vaccine escape and depending also on the mixing parameter. And the important thing is that then the ratio between different replicative values is quite simple, and that allows us to have analytical predictions for the selection gradient, which is quite simple, and tell us that the vaccine-adapted variant will spread faster if, of course, there is less control. Okay. If there is mutation arises in a highly transmissible strain, if the mutation causes a large effect in vaccine escape ability, but more interestingly, it's going to depend on the densities of the vaccinated and infected hosts in both classes of low or high contact. So what we can see is that for instance, the non-vaccinated hosts have no effect on this spread. Okay, what matters is how many hosts are vaccinated in the population. And what we see is that the contribution of the high contacts versus low contacts host is different, and there's a ratio m square between the two, which means that the evolutionary weight of hosts with more contacts is m square. So a twofold increase in contact number translates into a fourfold increase in evolutionary quality for the pathogen. Okay? So it's going to have a disproportionate effect on the evolution of the pathogen.
You said this is a quasi-equilibrium approximation.
Yeah.
This is an equilibrium of the epidemiological dynamics.
No, it's assuming that the reproductive value and class frequencies equilibrate quickly while the epidemic is still changing. So there's a kind of stabilization.
So it's the composition of the population that equilibrates fast.
Exactly, but also the reproductive value, so the quality of the different classes. But the R of the population, the growth rate, is still non-zero, so you still have.
It's still increasing.
Yeah.
And you have different S variables. I mean, S is the selection, that is the script.
Sorry, this is the session gradient.
Yeah.
And this is it's not the same, but it's true that we don't see. But this is the session gradient. It's not the stable host, but choice of adaptation. Just so a picture. So this is what we did. So we have a simulation where this is non-pharmaceutical intervention. So basically, we have a high control with lockdowns, and so you see that during this time there's no evolution of the virus. But then when we relax this, we have an increase in the evolution in the frequency of the vaccine escape mutant. And this, if there's no vaccination, of course, there's no increase. But if there's vaccination, you have an increase. And the speed of this increase depends on who we are targeting. If we target hosts with high contacts, okay, then we have a faster increase of the vaccine escape mutant. And if we target hosts with low contacts, we have a slower increase. And we can also look at the incidence and the number of deaths in the model. And the general conclusion that in this model, targeting hosts with more contacts speeds up the spread of the vaccine-adapted variant and also leads to a larger epidemic here and more deaths. So that was at least this result from the qualitative point of view. It suggests that the strategy of targeting people with low contacts and which are more vulnerable was good from an evolutionary point of view. But it's kind of a proof of concept model that you can potentially use this kind of models to try to generate public health guidelines to manage in real time an epidemic.
And what is the dotted line? Is that without?
The dotted line is without vaccination.
Oh, yeah.
So you see you have more death without vaccination, which is consistent given what we did. But you see also that you have a steeper epidemic if you don't have vaccination. The vaccination is still positive, but depending on which strategy of vaccination you choose, you have different outcomes, and some are better than others. But you could also have some situations where you predict, okay, maybe the selection for vaccinated virus is lower, but you have more death. And that's where it starts to be complicated if you take other assumptions. So at least in these assumptions, it's consistent with what people were doing at the time, but who knows what would happen in different models.
And with this result, all if you don't have the quasi-steady state approximation.
So one thing is that this quasi-equilibrium transformation, it was quite well. I think you see and plane line, and then they correspond to the simulations with and without this approximation. So it's quite good for the change in frequency. What we are still working on is it seems that we have used this approximation for other models, and it seems to work even in cases where it shouldn't, from kind of what we expect. And so it seems there's it's a kind of a very robust approximation in many cases, and we still don't really know why it works so well. Because it works even in cases where, because if you have periodic fluctuations, you shouldn't be able to do this quasi-equilibrium approximation. You should do something different, and still not bad. And so we are still trying to see if it's due to the scenarios we are looking at, where you will maybe have an epidemic and so on, maybe it changes things. And we are collaborating with some mathematician colleagues to try to shed light on this, because it's unclear why. Sometimes it's too good to be true that it works, so it could be that there's something we don't understand. But at least in this case, it works well, and so you can get some analytical insight.
So does this approximation actually then imply that the composition of your host population has stabilized, but for example, all people have higher death rates? So you mean, does it then imply that they already have died, and so their frequency has gone down, or something like that?
No, I don't assume that. It assumes that even if the population is still growing or changing over time, the frequency of the different classes in the infected compartment remain roughly constant. It's in the infected compartment, so the frequency of the pathogen in the different classes. So it's not the frequencies of the young or old people in the population in general. So only the eye are looked. So that makes a difference too, because it doesn't say anything about the stress structure of the population. And that's because we look at pathogen evolution. If we looked at the host evolution, we will need to take the whole. Okay, and so the concept of background here is that the reproductive values we are looking at are dynamical, which is often not what is assumed in many models in ecology, where the reproductive values are constant, okay, because we look at the long-term synthetic growth rate of invader, and so the reproductive value are, for instance, the left eigenvector associated to the domain eigenvalue that give the speed of this rate of increase. And so often it's a constant, okay, in most textbooks. Here it's dynamical, and the intuitive explanation that because the density of susceptible hosts in the different classes are not constant, we expect the quality of the different host class to change over time, okay? At a given time, a given class is not going to be as valuable for the pathogen as the next time. And so the idea that we can write equations for the change in the frequency of the mutant, but also the change in the frequency of the pathogen in the different classes, and the reproductive value of the pathogen in the different classes, which is V. And we have this kind of a joint system actually that we can solve using, again, assumption of separation of time scale, because especially if evolution is slow, then this is going to be a small parameter, and we are going to have a slow variables, and these are going to be fast variables. So we can, in principle, use a separation of time scale to calculate this V and U parameters that can then use in the. So this is U here. You can then use in our model to calculate the selection gradient. And in fact, one thing we can do is also use that to look at a long-term adaptation. So this was short-term adaptation, because as I say earlier, imperfect vaccine, they have the potential to select for increased pathogen virulence. One example is disease of poultry, which is Marek disease, where they have used vaccine, several types of vaccine over the years, and each time they introduced a new vaccine, there was an increase in the virulence of the pathogen. That's because the vaccines were vaccines that only removed the symptoms of the disease. So it was removing the virulence, but if you remove the virulence, there's no cost to increasing transmission. And so then the disease started as a pretty mild disease where chicken were not looking great in the market stalls, but still they were okay. And now it's kind of a weird cancer that is really very aggressive. And so it's quite documented that vaccines could have an effect on the evolution of this virus. So to understand what's happening, one thing we did is to look at what happens if vaccination coverage fluctuates in this kind of setup. And then the dynamical reproductive value approach I suggested before allows us to calculate the selection gradient, but that's now an average selection gradient over one period of the fluctuations. and you can get an approximation where you have a balance between selection happening in the naive class where there's no vaccine and the class of vaccinated host with a single parameter that controls which is the dominant process, and this single parameter is the average reproductive value of the vaccinated host over one period of the fluctuation. And that determines the balance between selection in naive versus vaccinated host. And so the idea is basically if you do this kind of model, you have fluctuations in vaccination coverage, you have fluctuations in the densities of the different hosts, and for the different two classes of naive and vaccinated hosts, on the attractor, on the periodic attractor, you have also fluctuations in the quality of the different classes for the pathogen. So and then you can use this to calculate the section gradient. And you see that the change of frequency is predicted with this approach is very consistent with the change, what you will predict using Floquet theory, which will be the rigorous way to, the exact way to deal with this periodic system. So it's a very good approximation. And the nice thing that you retain your interpretation in terms of reproductive values and densities, which are very important in evolutionary ecology, very important concepts. You guys move that, but the basic result of this model then was that if you increase the period of the fluctuations, the virulence, eventually stable virulence on the long term was decreasing for two types of imperfect vaccines. And same if you change the fraction of the period, which is with treatment, with vaccine. It's also compared to the case where you are, which is constant, where you have no fluctuations, you can have a mitigating effect of the negative effects of imperfect vaccines if you have fluctuations of the vaccine, which is not necessarily what you would expect, because if you think of antibiotic resistance, often people say, well, if you don't take your antibiotic during the 1 week and you have fluctuations, you start taking it and then stop, it's going to be bad for evolution of resistance. But here, for the evolution of virulence, it's kind of a mitigating effect, and that's potentially important because fluctuation like that, there could be like a crude representation of what happened when you have vaccine scares or when you have pulse vaccination strategy. So you cannot make predictions only using constant vaccination model. Okay, so that's the end of the part 2 and also the end of the talk. Sorry, I'm maybe a bit long, but you are very patient or too polite. I don't know. So the conclusion of this part is that to look at heterogeneous population, we can use a classical concept in demography and evolutionary biology, which is reproductive value, and we can actually use that to model viral adaptations to vaccines or apply questions. And so that's the interesting thing, that you can use general evolutionary ecology theory and apply it to understand more applied questions like what is the evolutionary impact of public health interventions such as vaccination. And what we are trying to do with my colleague Sylvain Grandon is to try to extend this to further biological realism, like such as age-structure, spatial structure, which is all different ways of adding classes to the population. That's all for me. So I've just put some reference for the two parts of the talk, and I'm happy to take any other questions if you have some. Thank you.
Okay, I have a small question. So
Yep.
can you please elaborate a little bit on this dynamic reproductive values? So because in my understanding, it mainly comes from this equilibrium states, right? But you were talking about the dynamic reproductive.
Yeah. So actually, if you look at this this equation, if you assume that this is stable, this is 0, so you can have you assume that this has stabilized. This is exactly the eigenvalue left eigenvector equation that we are used to. Okay. So the quasi-equilibrium as approximation I was talking about for the vaccine case is just assuming that this holds at each time. Okay. So that at a given time, you are always close to this agent vector, agent value problem, and you can solve it. And so you're just going to change. I think this is not, I mean, I think physicists do this kind of thing often. I'm not sure I master the mathematics behind it because I'm not a mathematician at all, but this is the idea. And then if you have a periodic attractor, then what you're going to say that on average, this should be zero on the average over one period, and we can also derive some analytical stuff. But now you're going to say that this right-hand side is zero on average, and you can derive also some relationships between different average quantities that can be used to simplify the section gradients when you have periodic dynamics. There's another one of the paper I have in the reference is actually to use that in the price equation. And the reason I did that it allows you to show that if you instead of looking at the mean, if you have a class structured population, you weight the mean trait in each class by the reproductive value at a given time. And then what you can do is write a change in the mean trait in the population that is only due to selection. So you remove the effects that are confounding, that are due to the fact that due to demographic transitions between classes and the fact that the classes do not have the same reproduction and survival vital rates, then you'll have some change in the mean trait that can be due to just transitions between classes and not to selection. So if you don't do that, if you empirically you look at the change of the mean trait, you have fluctuation, you could say, yeah, that is selection, but actually it could be a neutral trait that is just changing because of some demographic processes. So the reproductive value, they allow you to actually extract the signal of natural selection from what could be complex dynamics that is due to a combination of demography and evolution. So that's why they keep coming up in all the selection gradients in heterogeneous population, because that's what they do. They just allow you to focus on selection only.
Okay. Any other questions? I have a small.
Yeah,
I have a small. So in the first part of the talk, we were talking about increased virulence leading to increase in spread of the disease, right?
Increased virulence leading to
increase in the transmission.
Yeah, so that's an
So I was thinking like increased virulence, wouldn't it like kill the host population and then reduce the transmission? Thinking in the correct way?
Yeah, no, it's just what we assume usually in these models is the heart of the virulence evolution theory is that you have a trade-off between transmission and virulence, which have this form often, and the rationale that virulence is due to the within host proliferation of the virus, for instance, but transmission you need also to have within host replication. So when you increase within host replication, you also increase the transmission. Okay, you also increase virulence. But at some point, you just only have negative effect on the host, more than positive effect. And so the benefits in transmission are saturated.
So at some point it
saturates or it decreases. But this concave shape means that if you increase transmission, you often, in this case, going to if you increase virulence, you're going to increase transmission.
So what I'm thinking is more at the later part of
the later part, yeah. And so what happens in this starting model that the SS is sitting on this part because in the simplest model, you just draw the slope tangent to this point that goes to the origin, and so it's sitting in that and that's the marginal value theorem. So in this simplest model, you don't never worry about that. It's kind of irrelevant because you are selected for an optimum that is before this peak. So you often like that. But so if you look at only at this part, then an increase in virulence indeed leads to the increased transmission. That's a weakness of the of this is that's not very mechanistic. It's kind of an implicit phenomenological representation of what's going on. And if you want to do a more mechanistic model, you have to go into the within host scale, and then it's a kind of a black box, and you don't know, depending on the disease, things are very different within the host. You could model anything. You have to model co-infections. You have to model so many things that it starts an interaction with the immune system. So it's really a difficult problem. So most models, they take this kind of phenomenological approach where they use just two parameters, but it can be debated whether it's the best way to make progress for more realistic scenarios.
Okay, if there are no more questions, then we will let's thank the speaker once again.
Thank you.